Hi, okay. Today we are going to start Unit 2. In this, the topic is digestion and absorption of carbohydrates, proteins and lipids. Okay, now today we'll first start with the digestion of carbohydrates. So first let's see what are the different types of carbohydrates that are present in our daily diet. First one is let's say polysaccharides. Polysaccharides are nothing but poly means many, saccharides means sugars, polysaccharides refers to many sugar units. It's a polymer of simple sugars. And basically we have two types of polysaccharides that are available in our food. One is glycogen. These are non-vegetarian, present in non-vegetarian diet. Like they are actually the storage carbohydrates in animals, and starch is a storage carbohydrate in plants. So any plant source will have starch, and most of the animal sources will have glycogen, stored glycogen in them. Okay, so these two are two are polysaccharides that are available in our diet. Next one is disaccharide. Di refers to two, saccharide again sugar. So disaccharides refers to the sugars that are two sugar units. Okay, examples, sucrose. So when you are eating sugar, okay, so that contains sucrose. If you are drinking milk, it contains lactose, and the grains contain maltose. Okay, so sucrose is also called as a table sugar or cane sugar. Milk sugar is lactose, and grain sugar is maltose. All three are disaccharides. And then there is also monosaccharide. So monosaccharides are simple sugars, the simplest form of sugar that cannot be further broken down. Okay, and basically they are glucose and fructose, sometimes even galactose. In addition to that, there are other simple sugars like alcohol, lactic acid, pyruvic acid, pectins, dextrins, etc. Okay, in addition to all these three, different types of sugars, simple sugars or non-reducing sugars or complex sugars, we also have cellulose in our diet, but these cellulose cannot be broken down in our body, in our digestive system, because we lack enzymes to break them. So that's why they are not considered as a dietary component. These are structural polysaccharides. Okay. Now let's go on to our actual topic of discussion, that is digestion of carbohydrates. Now before going to the carbohydrates, let's see what is digestion. Digestion is a process of breaking down large molecules into smaller ones. Okay, there are two types of digestion: mechanical and chemical. Mechanical digestion means, okay, here physical breakdown. A larger molecule or a larger bite, okay, is broken down into smaller one. For example, you have taken a whole chapati, you can't eat, you make a small piece of it and then put it in the mouth. What do you do after putting it in the mouth? You chew it, isn't it? By using your teeth and tongue. This process is a physical breakdown of the food particle into smaller ones, and this is called as mechanical digestion. And that process of breaking down the larger particle into smaller ones by mechanical action of the digestive tract is called mechanical digestion, and in mouth it is called chewing, or also known as mastication. So it will make smaller pieces of food. Next one is chemical digestion. A chemical digestion means here certain chemicals are used, like enzymes are used. Okay, so this is a process of breaking down larger molecules, their particle, here molecules, into simple molecules that can be absorbed. I repeat, chemical digestion is the process of breaking down complex molecules into simple absorbable forms. Okay, and here it is generally done by the help of enzymes. Remember, chemical digestion occurs throughout your alimentary canal. Now we are talking about carbohydrate digestion only. Okay, rest of the types of digestion we will see in our next class. Now, in mouth, we know that salivary glands are present and they secrete saliva. This saliva contains enzyme called as salivary amylase. Okay, this is the amylolytic enzyme. Lytic refers to breaking some amylo compounds, that means carbohydrates. Okay, so in mouth this enzyme is called salivary alpha amylase, or also known as taline. Okay, now this is active in pH of 6.7. that's the optimum. Anything plus or minus with this is okay for its activity. Okay, and this enzyme still remains active even in gastric juice because its active sites are protected. Okay, now in mouth, what happens when you take a food that contains carbohydrate? This amylase enzyme, for example, let's say that you have eaten starch. Okay, what happens? The starch is formed of amylose, a linear chain of glucose, and amylopectin. Amylopectin is a branch chain. These long chains of glucose are formed of alpha 1,4 linkages. So linear linkages are alpha 1,4, branching occurs by alpha 1,6 bond. Okay. Now amylase can break alpha 1,4 bond, but it cannot break alpha 1,6 bond. Again, this won't break the terminal bonds. These terminal bonds will not be broken. As a result of this, what happens? Maltosis, maltotriosis, glucose oligomers, and alpha-limited dextrin is formed. Alpha-limited dextrin is something that contains a branch chain. Okay, so these are different compounds that are produced as a result of activity of amylases or alpha amylases. Okay, now that food, when you swallow by a process called deglutition, will pass through from the mouth to the stomach through esophagus by a process called peristaltic movement. Okay, and peristaltic movement also adds to some sort of mechanical digestion. But stomach doesn't contain enzyme for the digestion of carbohydrate, so there is no digestion of carbohydrate in the stomach. Next, move on to the intestine. So the intestine, the first part of the small intestine is duodenum. Food from stomach goes to the intestine. At this time, what happens? The intestine receives enzymes and secretions from both liver and pancreas. Not only that, it also has its own glands. So secretions of liver, pancreas and intestine meet up in the intestine, and they contain different enzymes and salts. Now, we are concentrating on carbohydrate. So, the amylolactic enzymes are derived both from pancreatic juice and succus entericus. Secretion of intestine is called succus entericus. So, this amylolactic enzyme present in the pancreatic juice is called pancreatic amylase or amylopsin. Now, both these, salivary amylase that was present in the mouth and pancreatic alpha amylase present in the secreted by pancreas present in the intestine, can hydrolyze alpha linkage 1,4 linkages which are internal, but it cannot break alpha 1,6 linkage and 1,4 linkage. Just like salivary amylase, pancreatic amylase also can break only alpha 1,4 linkages. Those are internal ones. So as a result of this, at the end of pancreatic amylase activity, once again you see that there will be maltose, maltotriose, glucose oligomers will be formed in addition to alpha limited dextrin. Okay, so these are the products of breakdown of polysaccharides in intestine as a result of enzymes secreted by pancreas. Now I said, in addition to pancreatic amylase, intestine also receives enzymes from its own glands, and these enzymes are maltase, sucrase, lactase, dextrinase, trihalase and isomaltase. Isomaltase is responsible for hydrolyzing alpha 1,6 linkage. In our previous slide we have seen this linkage will be broken down by isomaltase. Okay, and the lactase will break down lactose into glucose and galactose, trihalase into will break down trihalose into glucose, sucrase will be broken down in sucrose will be broken down into glucose and fructose by sucrase, and maltose will be broken down by maltase into glucose and glucose, because maltose is formed of 2 glucose units. Okay, so this is what you see. This is what we have discussed in our previous slide. So this isomaltase will break down this, and it will produce a maltotriose, and either isomaltase or maltase can break it down and produce glucose molecules. Okay, now just let's see the summary of the digestion. So in mouth, disaccharide and dextrins are produced as an end product. In stomach, there is no carbohydrate digestion. Pancreatic juice, that is a pancreatic amylase, the end product is dextrin, maltose and maltotriose. And as a result of enzymes of succus entericus, you see glucose, fructose, glucose, galactose are produced by breaking down sucrose, maltose, lactose, etc. So that means the end product of this digestion is glucose, fructose and galactose. Recall, chemical digestion is the process of breaking down complex molecules into absorbable simple forms. Once again, here you are seeing glucose and fructose and galactose are produced as a result of breaking down starch or glycogen or even the sugar present in your sugar or fruit, the sugar present in the milk and even in the grains. Everything is finally converted into the most simple form that can be absorbed, that is glucose, fructose and galactose. Okay, so by the end of the digestion, chemical digestion, these are the three sugars that are available. Now they'll have to be absorbed. So what is absorption? Absorption is the process of transporting the simple forms of the nutrients across the membrane towards the circulatory system. It is the process of transporting the nutrient from the lumen of intestine to the circulatory system. That is called absorption. And we know that there are three such nutrients that has to be absorbed: glucose, galactose and fructose. Now first let's see glucose and galactose absorption. This glucose and galactose absorption takes place by a process called as co-transportation. Co-transportation refers to a transport system where two different types of molecules are transported through or across a membrane. Okay, and here it is sodium that is a transporter of glucose or galactose. Okay, now this happens by the help of special transporters that are present in the membrane. So this, what you are seeing here, is the intestinal cell that is present in the outermost layer of the intestinal wall, and this is the lumen where the digestion is taking place. Okay, and this is the submucosal region, a part of the intestinal tissue, and here there will be blood capillaries. So the food from digested food from here should go to this part. Now how it is done? See, here is this transporter which is known as sodium glucose transporter. So co-transporter, and it takes sodium and glucose at a time. Now this process is slightly complex because the cell contains sodium. This sodium will be first pumped out of the cell, okay, and to balance that, potassium will be pumped in. This happens by the help of a special protein complex called as sodium potassium ATPase. Okay, this is the energy utilizing step. So sodium will be pumped out. Now what happens? Sodium concentration is less here. So sodium can now move from lumen to the cell, but it cannot happen directly. It happens by the help of this transporter. So this sodium binds here. As soon as sodium binds, some conformational change happen and sodium moves from here to here. But before that, because of the conformational change, glucose and galactose will bind, and then both these are transported across the membrane into the cell or into the cytoplasmic area. Once they are inside, now sodium again will be pumped out by the help of sodium potassium ATPase, and glucose will be transported out through another transporter called as glucose transporter 2. Okay, so this glucose will then go to the blood. And here you see the glucose, sodium is moving from a region of its higher concentration to a region of its lower concentration. Glucose is moving against its concentration gradient. Both glucose and galactose can be passed in the similar way. Whereas there is a last one, the fructose. This is transported through facilitated diffusion. A facilitated diffusion is a process where it is different from simple diffusion. In simple diffusion, what happens? The molecules will be transported from a region of their higher concentration or to a region of their lower concentration without any special help. Whereas facilitated diffusion can happen by the help of special protein molecules. They will help in moving across the membrane. Okay, so fructose movement is by the help of such transport protein. Here it is glucose transporter 5. Okay, there are 14 such glucose transporter in different organs. In the intestine, we find two types, glucose transporter 5 and 2. So here fructose is facilitatedly diffused through this membrane, and it enters to the cell, and from here it is passed out through glucose transporter 2. So glucose transporter 5 and transporter 2 will help in passing the fructose to the blood vessels, to the capillary. From here it is transported to rest of the part. Okay, so that's about the absorption of carbohydrates. So that ends our class. The summary of this lesson is human diet contains 3 types of carbohydrates, and digestion of carbohydrate takes place in mouth and intestine. Amylase, sucrase, lactase, maltase are major enzymes of digestion, and absorption of carbohydrate takes place by the help of active transport system and facilitated diffusion. These are my references for today's class. Thank you.